Hello everyone, this is Professor Dr. Satish Polshettiwar. Let us discuss today understanding of viruses. In today's session, we will be exploring structure, types, properties, transmission method, diagnostic techniques, how we can cultivate the viruses and treatment option available. Let's watch this video. Thank you.
Welcome to Dr. Satish Polshettiwar Educational Channel, your destination for in-depth and simplified pharmacy and life sciences education. Viruses. Structure, types, properties, transmission, diagnosis, and treatment. Viruses. Tiny but mighty entities are responsible for some of the most notorious diseases known to mankind. But what exactly are viruses? Let's dive deep into their fascinating world. Viruses are microscopic infectious agents that can replicate only inside the living cells of organisms. They are considered obligate intracellular parasites, meaning they need a host cell to survive and multiply. Typically, a virus has three main components. Nucleic acid core contains the genetic material, either DNA or RNA, but never both. Capsid, a protein shell that encases the nucleic acid. Envelope, in some viruses, a lipid membrane derived from the host cell, containing glycoproteins that help in host recognition. Viruses vary greatly in shape from helical and icosahedral to complex bacteriophage structures. Viruses can be classified in multiple ways, commonly based on a genetic material. DNA viruses, for example, herpes virus. RNA viruses, for example, influenza virus HIV. B. Presence of envelope. Enveloped viruses, for example, SARS-CoV-2. Non-enveloped viruses, for example, adenovirus. C. Host range. Animal viruses. Plant viruses. Bacteriophages. Viruses that infect bacteria. Viruses are unique in that they are non-living outside a host, but living inside. They lack cellular organelles, metabolism, and energy production. They exhibit host specificity. Can mutate rapidly, especially RNA viruses. Can't be killed by antibiotics. Viruses can spread through airborne droplets, for example, influenza, COVID-19. Direct contact, for example, herpes, HIV. Vector-borne, for example, dengue via mosquitoes. Fecal-oral route, for example, hepatitis A. Blood transfusions and sexual contact. Early and accurate diagnosis is key. Common methods include PCR, polymerase chain reaction, detects viral DNA RNA, rapid antigen tests, serology checks for antibodies, electron microscopy, research level, culture methods for specific viruses only. Treatment depends on the type of virus and the disease. Antiviral drugs, for example, acyclovir, oseltamivir, symptomatic treatment pain relievers, fluids, vaccination highly effective for diseases like measles, hepatitis B, COVID-19, preventive strategies, hygiene, safe practices, vector control. Let's revise. Viruses are tiny infectious agents. They consist of genetic material, a capsid, and sometimes an envelope, spread through various routes and need a host to multiply. Diagnosed through PCR, antigen, and serology tests. Managed through antivirals, vaccines, and prevention. If you found this video helpful, don't forget to like, share, and subscribe to Dr. Satish Polshettiwar Educational Channel. Stay tuned for more simplified science and pharmacy concepts. Drop your questions in the comments below. Empowering knowledge, inspiring future pharmacists.
If you found this video helpful, please subscribe, like, and share it with others who'd benefit. See you in the next video.